Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is meant for 7th August 2020, and our memory as usual helps us in tracing out what we have been studying, reflecting upon, understanding and making conceptualization. And in our previous session we were studying about how exactly photosynthesis takes place, what are the intricacies involved in photosynthesis. Though in junior classes we have been writing that overall photosynthesis can be represented as CO2 plus H2O, like CO2 plus H2O, and in the presence of sunlight and a pigment called chlorophyll, you get a monomer C6H12O6, right? And this is becomes starch when it becomes N times, and as byproducts some water will be formed as byproduct, plus most important oxygen will be released, which we are all enjoying free of cost. So this is what we have been writing in junior classes, but I said that in reality you understand now after having listened to yesterday's session that CO2 will never combine with H2O, and also I hope you understood that the oxygen that is getting released is due to splitting of H2O. It is the H2O that splits and oxygen is released, because there was a dispute and discussion whether the oxygen that is released at the end of the photosynthesis, is it because of the split in H2O or because of split in CO2, because in both O2 is there, CO2, H2O. Now the latest research shows, and it is proved beyond point, that it is hydrolysis or photolysis that results in release of oxygen. And now the question is, what are all the electrons that are released? That is E minus, we said yesterday, if you recollect from your memory, we said splitting of water. Look at this, yesterday I told you sorry, the H2O molecule, one molecule if you take the splitting of photolysis, H plus and OH minus, another H2O molecule, H plus and OH minus, another H2O molecule, H plus and OH minus, another H2O molecule, H plus and OH minus. So this is how water molecule splits. And when water molecule splits, what are the byproducts? It is 4H plus. You can see 1, 2, 3, 4. So 4H plus. And then the H2Os. So you have got one molecule of H2O and another molecule of H2O, so 2H2O. And now one oxygen and another oxygen, O plus O, O2. That's how we say O2 is formed beyond doubt because of photolysis, splitting of water. And what is left out in this reaction? That OH minus we are writing, because when water molecule splits, H2O splits, it is H plus and OH minus, and that OH minus, that minus indicates electron, and that's how this 4 electrons we are counting. What is the fate of this 4 electrons? What is the role that these electrons play? And this is how the electrons will play a crucial role in a process called photophosphorylation. So what is photophosphorylation? It is defined. Photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate. So we write adenosine diphosphate ADP plus 1 IP inorganic phosphate. So that results in formation of energy molecule adenosine triphosphate. And I said this is the currency of the cell. ATP is the currency of the cell. So the formation of ATP, because why photophosphorylation? Photo means light. Because of presence of light, what is happening? Phosphorylation. Inorganic phosphate is combining with ADP. And of course further you can know it is actually adenosine monophosphate, AMP will be there, and this will be having one IP, inorganic phosphate, that becomes ADP. ADP plus inorganic is ATP. That's how we are saying phosphorylation. So the process of formation of ATP with the help of sunlight can be a photophosphorylation. And also it is not only ATP that is formed. Here you can see there is a formation of NADPH. And again, if you recollect from your memory, this NADPH, the high energy molecule, is formed in PS1. If you recollect from your memory, I said there are two complexes, PS2 and PS1. And the PS1 has got P700. 700 is the nanometer wavelength, and PS2 is 680 nanometers. So in PS1, if it is ATP that is formed, in PS2 it is NADPH that is formed. Okay, so just read this, then we will go back to that cyclic and non-cyclic. So the process of formation of high energy chemicals ATP and NADPH in the presence of light is photophosphorylation. And phosphorylation is the process by which living organisms extract energy from oxidizable substances and store this in the form of bond energy like ATP. That's why we say energy currency. ATP is the energy currency of the cell. So phosphorylation takes place inside the cell, mitochondria as well as chloroplast. So it is basically of two types, cyclic photophosphorylation and non-cyclic photophosphorylation. Why is cyclic? Cyclic photophosphorylation, electron gets released, okay, and it gets excited, goes through the route of PS1, that is P700, and reaches to a substance called ferridoxin and makes the ferridoxin get oxidized. As a result of that, the NADP plus H plus will form NADPH. And the electrons that are released in the process, ferridoxin will put back that electron to the electron transport chain, particularly cytochrome B. So from cytochrome B, it goes to again PQ, various enzymes like cytochrome F, and back it comes back to PS1 complex. So it's a cycle. That's why we say cyclic photophosphorylation. Okay, and non-cyclic is the jet scheme which I explained to you yesterday. That is both the centers, light centers are involved, PS2 as well as PS1. So the light harvesting center in non-cyclic photophosphorylation is, it starts with the P680. From P680 it goes to electron transport chain. From electron transport chain it goes to PS1. From PS1 it goes to ferridoxin. So the electrons are not coming back to P680. Since it is not a cyclic form, instead it is in jet shape, we say it is non-cyclic photophosphorylation. And what happens in this? The difference between non-cyclic photophosphorylation and cyclic photophosphorylation. So two photosystems work in series. First it is PS2 and then PS1, then only the jet scheme. So these two photosystems are connected through electron transport chain, that is in the shape will be Z shape. ATP and NADPH plus H plus are synthesized by this process. PS1 and PS2, the PS1 is P700, PS2 is P680, both are involved. And where does this take place? It takes place in the grana, particularly lamellae of the grana. Please remember this, UPSC ND exam. Hence this process is carried out in granum, that is inside the chloroplast. If you recollect structure, I told you to draw the diagram, but so far I have not received, not a single boy has sent me the diagram. I told you to draw, but nobody has done it. Please do it. And in cyclic photophosphorylation, it is only PS1 works. In non-cyclic, both PS2 and PS1 both will work, whereas in cyclic only PS1 works. And the electron, because it is cyclic, it should be circular shape. So the electron circulates within the photosystem. That is how it happens in the stroma. Look at that, cyclic will happen in stroma. What about non-cyclic? Grana. Remember this. And because in this region PS2 is involved, photosystem 2 is involved, and NADP reductase enzymes are absent. So in cyclic photophosphorylation there is no involvement of PS2 and there is no NADP reductase. It is only PS1 works, photosystem 1. That photosystem 1 is, as I said in my previous classes, it is P700 and PS2 is 680. We say P680. So it is not involved in cyclic and hence only ATP molecules are synthesized. So just put a table, write differences between cyclic photophosphorylation and non-cyclic photophosphorylation and write the differences. Then you understand better. Just to make you further clarity, to make you understand better, let me take you back to what I said yesterday. That is jet scheme, and we were talking about this jet scheme PS1, PS2. Yeah, this is what here. This is what I want you to draw again. Now, we said the cyclic photophosphorylation. Cyclic photophosphorylation takes place here, P700 only. Now, what happens is when the electrons get excited, so from electron transport chain, the electrons are coming. And these electrons moving further reach to the electron acceptor and then it goes to a substance called, I said, ferridoxin. I can write it as EFD, ferridoxin. So the electrons reaching ferridoxin makes the NADPH formation. How this NADP plus, I said yesterday, H plus. What about the H plus that is there? That is this one. Look at that. So during the water splitting, hydrolysis or photolysis, we were talking about the fate of electrons. What about the fate of H plus, protons? Protons are leaks. So NADP plus H plus will make NADPH. So this is highly energetic compound. And in the bargain of formation of this, the ferridoxin gets excited and again electrons are released and these electrons will go back to the chain. So you got a circle. So from ferridoxin they come here, again to LH center, again to electron acceptor, again it goes to ferridoxin to NADPH, again back. So this is how. So we say cyclic photophosphorylation. What about non-cyclic photophosphorylation? Both LHC P680, PS2 as well as PS1 is involved. So here the electrons get excited here, so light harvesting center, and these travel to electron acceptor. Varieties of cytochromes will be there, cytochrome A, cytochrome B, cytochrome F, like that, and all this will be, electrons will be reaching this way, and here ADP plus ATP will be synthesized. So in non-cyclic photophosphorylation, there is involvement of PS2, photosystem 2, electron transport system, as well as PS1. So PS2, ETS and PS1. And finally, since it is in the form of jet shape and the electrons that are released here, they are not coming back here. Hence it is not cyclic, it is non-cyclic. Hence there are two systems that take place. That is cyclic photophosphorylation, where PS1 is involved alone, but it has got very good benefit. What is the benefit? NADPH are synthesized. Whereas in non-cyclic photophosphorylation, both are involved. Photosystem 2 as well as 1 is involved and ATPs are synthesized. Hence, the purpose of photophosphorylation is production of energy-rich